Thank you for the kind introduction. So I'm just going to follow, my previous speakers, talking about a higher order correlation model to understand the epigenetic microenvironment in breast cancer. I'll take slightly, different approach, but it's really within the same domain. So briefly, I want to say that my lab studies epigenetic circuitry. Just a second. I'm not, okay. Sorry, I'm a Mac person, so I was like,
You could just use the arrow keys.
oh, just the arrow keys. Okay, great, thank you. My lab studies epigenetic circuitry of cancer cells by investigating the roles of histone modifications in gene regulation. So the major questions we have is to, we're interested in understanding the molecular mechanisms that guides the choices of about the cell fate and connectivity during the cancer initiation and progression. And for this talk, I really wanted to talk about, how this regulatory signals from where they reside in the microenvironment can contribute towards the cancer formation. So to kind of get into understanding of tumor formation, we have to understand what is the microenvironment. So typically in adult tissue generation, there are maintenance that are mainly regulated by continual turnover by mature cells. And this process is mediated by presence of tissue-specific stem cells that functions depends on both intrinsic and extrinsic molecular signals. And this adult stem cells resides in this, sorry, I'm having a little trouble clicking on this. Well, sorry about that. Well, I just want to go into, sorry, how do I actually go into this area? Oh, just okay. Okay, great. Thank you so much. They reside in specific tumor microenvironment that are composed of those two components, cellular components and the non-cellular components. And the cellular components are usually comprised of stromal fibroblast, mature cells, immune cells, adipose and endothelial cells. Whereas the non-cellular components are this extracellular matrix, growth factors, and cytokines. And normally when the microenvironment, there's an alteration or disruptions in microenvironment, it affects the normal tissue morphogenesis, which then promote the cancer formation and progression. And that, and when it turns into a tumor, this tumor interacts with same, within the same microenvironment, and the process we call it is the tumor microenvironment. So extracellular matrix component plays a very important role that constitute up to 60% of the tumor mass. And it can be viewed as a network of interactions of proteins, macromolecules that participate in various cellular processes such as proliferation and cell migration and growth. It plays a really important role in tissue structure because it provides the support and the maintenance of tissue architecture and nutritional support to the surrounding cells. And ECM, the dynamic of ECM is that it interacts with all these different growth factors and cytokines to really mediate the cellular behavior, contributing to all this growth, progression, and differentiation of the cells. So typically, when we think about tumor microenvironment, most of the studies have concentrated on looking at immune cells and their interactions with other component or the microenvironments. But recently, we are seeing that, particularly in this study, where there are contributions from epigenetic marks. So specifically the histone mark. In this particular study, they have looked at EZH2, which is a subunit of a polychrome repressor complex that mediates certain histone marks to, change the chromatin environment. However, if you look at this particular, so they looked at the H3K27 trimethylation, which is a repressive mark of histone that is well studied among a lot of diseases as well as cancers. They're just a handful of the canonical ones that are available, whether it's in TCGA or ENCODE. There's much more connectivity that's going on and interactions with various different combination of different histone marks. So what we're trying to do in this particular study is to strive to understand the complex interactions among molecules and the tumor microenvironment, looking at various different combinatorial effects of histone marks by looking at the regulation of signaling proteins and this various histone marks when they're treated after the growth factor ligands. And so we're hoping that the expected results would give us an idea of onsets of specific protein histone signatures in response to this growth ligands, and hopefully that will allow us to understand the initiation that occurs during the breast cancer formation. I don't have to actually explain a lot about the chromatin reorganization. I'm sure this audience is very well aware of it, but I wanted to just bring in the component of the histone marks, how they regulates the packaging of chromatin by going through this post-translational modifications through various different biochemical modifications where acetylation, methylation, ubiquitination, or phosphorylation could happen through enzymatic processes. And why is that important? Because whenever we have these different marks at the histone tails, it actually confers to a unique chromatin fingerprint. So for an example, H3K27 trimethylation is a repressive mark that has shown a lot of impact in a lot of diseases, including cancer. H3K36 trimethylation is an active mark. So, but what happens when they co-localizes, giving rise to this bivalency idea? We have no idea how that works. And why is that important? And that's important because there's a process called chromatin remodeling, where changes of nucleosomes or position or mediated by this various different histone marks and other chromatin remodeler proteins such as the reader, writer, and eraser really work in a very tightly coordinated manner to in regulation and transcription machinery. So to address that problem, a large-scale initiative, the Library of Integrated Network-Based Cellular Signatures, LINCS, sponsored by NIH, has started this project called ECM, Extracellular Matrix MCF10A, which is the normal breast cancer tissue, not cancer, but the normal epithelial breast tissue, molecular deep dive to understand. They have carried out a multi-omics characterization of various breast epithelial cells of 6 growth-promoting ligands through capturing proteomics, genomics, and imaging data at various different time points to understand the signal, regulatory signals network within the change of the breast tissue morphology. So how are we going to understand the changes that we see in the structure of ECM? So we have taken the systems approach to sort of think of all those growth factors, which are the ligands that will be changing the genomic or the proteomics measurements by encoding all of those different assays into encoding, decoding, if I could say, a method, an algorithm that allows us to sort of take multi-omics approach to map them at a very various different time points to capture the dynamic. And hopefully, using that dynamic, we would be able to output the kind of information that will allow us to understand how they're mediating different kinds of cellular phenotypes. And so typically, interactions are represented as a network encoding pairwise correlations between nodes, and optionally with some mathematical formulation of the dynamics. Usual mathematical formulations of dynamic typically assumes that each node in the network has an associated state variable and its function of time. But what it doesn't do is it does not capture this multi-interactive component that tells us whether or not, so for an example, if you have a node A and D, it is associated with some kind of weight or the correlation that will tell us, how they are correlated. So this kind of model really doesn't work when we have multi-omics data where we're trying to map and understand how, at the time T1, T2, this dynamics is changing. What we have proposed here is we developed a higher order correlation model where this is a triplet, I can say, the relationship of ABC that accounts for, it spits out a score that tells me that W1 is a score among these three nodes, as opposed to just A and B or A and D. And the motivations for using the tensor, to construct the signaling proteins, ligands, and time three-way tensors, was the fact that it actually ensured the uniqueness that we always see that as a computer scientist, making it more robust. And this is the kind of this is a method that is sort of came organically evolved from the non-negative matrix factorization or the 2D type of matrix factorization or the factorization talk that we just heard. So how are we going to do this? So HOCMO is, it applies a tensor on the RPPA, which is the phosphoprotein data, and we'll call that as a regulatory signaling protein data, and GCP, which is a global chromatin profile of various histone marks that are assayed using mass spec. And so we represent those data into a tensor representation where you can see that there are 279 phosphoproteins with different ligands at different time points. So the workflow is that we would initially bring in all this multi-omics data, do some statistical significant test by performing differential analysis, and then we will use non-negative tensor factorization technique to really extract the latent factors that will allow us to understand how those latent factors could give us information about the inter-pathway signaling versus the correlation patterns, which can be derived from using the latent factors to look at the intra-signaling pathways. And hopefully with those, we would be able to understand the correlation among the ligands, and in this case, ligands, histones at different time points induced by different growth factors. So here are the steps. I think I should be going in a little bit faster, but I just wanted to kind of go through the methods and the material steps that we have taken. So we have taken 295 reverse phase protein array and mass spec based 64 different histone marks, combinatorial marks. And then the condition is that, we induce them with all these different 6 ligands. And these ligands were actually chosen because they actually are particular in promoting, changing the cell morphology and progression at different time points. And then we performed significant tests with respect to the control here, the PBS, and calculated the fold change. After we got the differential readouts from those data, we would then use the multi-omics data tensor representation to create them, and all of this is done in just one input part of the algorithm. And then we use this higher tensor concept of the tensor, just to kind of give you an idea of what tensors are. These are mathematical objects that generalize, scalars, vectors, and matrices to higher dimension. And one of the important aspects of it is that if you have a higher organization, tensor-based representation of tensor, you can actually break it down or decompose it into rank 1 tensor, because that allows you to be able to perform this computation using an outer product rule. And that's what exactly what we did. So in the next step, we applied the non-negative tensor factorization method that's called CanDecom PairFact, CP. And we chose this compared to the Tucker model only because it allows us to have this rigor, allow us to actually generate a unique solution, meaning it avoids the kind of rotation problem that you would see in matrix factorization or even in the Tucker model. So the idea is that you have a tensor, you decompose it into their latent factors A, B, and C. And in our case, this could be our histone, this could be the time and the ligand. And using those, and there's an R given a parameter controlling the number of rank of 1 tensors, we then use this objective function where you minimize the tensor in respect to all the latent, the summation of the latent factors that you see. And of course, we impose the non-negativity constraint here. And so to determine the optimal number of components, rank K, which is oftentimes is a challenge because you don't know how many functional modules or what are the clustering, type of cluster that you're going to get, we applied this core consistency metric to allow us to agnostically just look at through the data and learn and come up with the appropriate K. Then we can just put in and allow that decomposition to happen. And then the last step, we calculated the higher order correlation score using this triplet relation that we would get from the latent factors and then augment them into what we call the correlation matrices. Okay, so here are some of the runs that we did using about like 500 runs to with different K to see which one are the optimal K that we can find. And when we do that, we look at the distribution and we see that the CC and many other studies have shown that if you have performed 60% over, if your performance is over 60%, that's a really good CC. So from both diagrams where you can see both, this is for the histone and this is for the phosphoproteins, the K, the optimal K was 2. So using the optimal K equals 2, we actually were able to look at the latent factors. So you see certain signatures, and we were able to perform further clustering to kind of showcase that the distribution of each histones or the protein here, signaling proteins, how they look. And from the augmented matrices, which were generated by looking at the latent factors that we got from A and B, which is essentially tells us the interactions between the here would be the phosphoproteins and ligands, and then here the ligands at the various time points, and then sorry, this is the phosphoproteins with the time points and ligands and time points. And we see that there's an enrichment at 24-hour, suggesting that there might be, and we see the same thing in the histone level as well, suggesting that perhaps the cancer initiation that we're seeing is perhaps starting at the 24-hour when these growth factors were induced. We went on performing the selections of significant ligand, phosphoprotein, and histone triplets. So we've calculated all the HOC scores for all 274 phosphoproteins as well as the 68. And then, oh, I'm going over, yes. Sorry about that. And then we were able to use this elbow specific algorithm to actually point it out the handful of one that we really felt that was significant. And so we were able to construct a 3D kind of network here where you can see that by inducing various different growth factors, here are the phosphoproteins that are significant and how they're mediating specific histone marks that would be giving us whether it's a repressive or activating or repressing the DNA accessibility. So from there, we were able to identify. Am I have to?
Yeah, we're a couple minutes over already.
Right, sorry. So from this.
Do you have any final slide you wanted to show?
Yeah, this is the last one. So I don't know.
Acknowledgements. We're quite over.
Oh, sure. Okay, so these are the results that we found. We found some really potential biomarker that we are currently experimenting and doing validation. We look at the chip-seq that corroborates with our proteomics data sets. Same thing with the gene expression that corroborates with our time. And in conclusion, I would say that we're trying to use this to extend this work into single cell analysis to look at the in vivo environment, and hopefully we will be able to use the same model to understand the interactions in higher order modes. So I just want to thank my labs and NIH links, and here's the code for the GitHub that's available, and thank you for your attention. I'm so sorry it took up so much time.